So today the focus of the talk that myself and then Svetlana will cover is insecticide resistance and using red-legged earth mites as a case study to talk about why resistance is a potentially looming threat, and perhaps using that as a bit of an argument as to why we might want to be a bit more smart and strategic about pest control. And I guess that leads nicely into Laura's talk, who'll be talking about a national GRDC program looking at integrated pest management. The work that I'm going to be talking about today is a collaborative effort. There's a number of different researchers that have been involved, particularly from the Department of Ag in WA as well as from the CESAR University of Melbourne group. So there's a whole suite of people there that have contributed significantly to this work. So I'm going to talk a little bit about red-legged earth mites. Many of you in the room will probably know that they're quite an important pest within Australia. They're also found in New Zealand and South Africa, and they were actually introduced to Australia from South Africa in the early 1900s. They're widely distributed across southern Australia. They're found across the wheat belt of WA, South Australia, Victoria and southern New South Wales. Although this figure here is a little bit sketchy, it is thought to be fairly accurate in that the damage costs and application costs caused by red-legged earth mites are in the vicinity of about $500 million per year to the Australian agricultural industry. And management of red-legged earth mites can be complicated because they are often confused with other mites such as blue oak mites, amblyopia mites and even Balorsti mites. It's widely believed that the red-legged earth mite is responsible for more insecticide applications than any other invertebrate pest within Australia, and that does include things like diamondback moth, helicoverpa and aphids. What are we currently doing around control for the red-legged earth mite? I guess the story is fairly similar to the way we tackle a lot of broadacre grains pests, in that we are heavily reliant on broad-spectrum chemicals. Often these chemicals are applied prophylactically, so insurance sprays are still quite a common thing and a common practice across southern Australia. There have been some attempts at introducing biological control agents, and these have mostly been focused around predatory mites, such as this one here, but I guess largely you would have to say that they haven't really met with the anticipation and the expectations that they come with. Some people, some growers certainly do rotate their crops as a way of suppressing mite populations, and there's also practice of managing pastures in spring as a way of, again, building down your red-legged earth mite numbers in those pastures, particularly before going into a susceptible crop like canola the following year. In more recent times, there has been a push and certainly an adoption of insecticide seed dressings to combat a lot of crop establishment pests, including red-legged earth mites, and also the time right package that was produced by CESAR and AWI some time ago has also taken off somewhat over the last decade, but both these methods still are heavily reliant on insecticides. So if we just take a step back to some work that was done in the mid to late 90s, this is a paper by Hoffman and colleagues that was published in 97. In this work, they essentially identified evidence of tolerance to insecticides in the red-legged earth mite in Victoria, and from this paper they speculated or predicted that insecticide resistance would eventually evolve in red-legged earth mites. I guess from that point in time, you know, the situation really didn't progress any further for about 10 years, but as many of you will know, that about 4 or 5 years ago the story I guess evolved. And that was really this issue around insecticide resistance that evolved within red-legged earth mites. And when we actually look back at the history of controlling red-legged earth mites within Australia, and we look at this value here of the fact that we've been throwing chemicals at red-legged earth mites for over 5 decades within Australia, and given the high population densities that you find in the field with this particular species, it's probably not surprising that we are now faced with this particular issue. Certainly, you know, there's a hell of a lot of selection pressure that we've been putting on this species. So if we just briefly talk about the actual incidents where we had first confirmation of insecticide resistance in red-legged earth mites. Essentially we had a farmer near Esperance in WA who had a chemical control failure in an emerging canola paddock. The short of the story is that over a period of about 3 weeks, this particular farmer applied 4 separate applications of synthetic pyrethroids. The third and fourth were actually with mixtures of synthetic pyrethroids and often at higher rates than what are registered, and this particular farmer could still not get control of that particular mite population. Not surprisingly, he lost that entire crop and needed to resow that paddock, which I think from memory was about 200 hectares. If we look at the paddock history, it was really not too different to a lot of the practices that are going on. There was repeated applications of SPs for at least 5 years within this paddock, and no real evidence of chemical rotation over that period. So what we did working with the Department of Ag is to actually screen these mites and actually try to ascertain and elucidate what was actually going on. So these are some results for some pesticide bioassays that we conducted, and on this axis we have mite mortality, and this is pesticide concentration. In this instance we're actually looking at Telstar, bifenthrin. And what you can see here is a dose response curve for a susceptible population from Victoria, so a population that we call a control, and the dose response for the mites that we collected from Esperance. As you can see, that dose response curve is significantly shifted to the right, and this was, I guess, our first indication that we actually did have insecticide resistance within these mites. What we actually did with these mites from Esperance is to actually culture them in a laboratory and rear through the offspring and then test the second generation of mites. And that's this graph here, and again, the results are almost a mirror image of what happened at the adult stage. So, this indicated to us that indeed we did have resistance, and in fact that resistance was heritable. It was actually passed on to offspring. So we kind of knew at that stage, back in 2006, 2007, that this resistance was going to persist in the field. What we also identified very early on is that the levels of resistance that we were seeing within these mites was extremely high. So this is just some data that we published from this finding, and basically what we could do from the dose response curves is calculate LC50s, and then from there calculate a resistance ratio. So for that dose response curve that I showed you previously, when we actually calculate a resistance ratio, we get an extremely high value here. We get a value of around or over 200,000 times. That's a very high level of resistance that's found. And typically you don't see too many cases in the field where you get this level of resistance in insects, but even within other organisms such as weeds. What we also found was that when we tested that second generation, again, we had a very similar level of resistance being found. So with this level of resistance, it's certainly not surprising that this farmer had 4 chemical control failures in a row and didn't get any successful control of those mites. When we tested a second chemical, fast-tac, alpha-cypermethrin, again, we found a very high level of resistance when we calculate the ratios of about 60,000-fold. The good news was at that point in time, though, that we looked at O-methoate, LEMAT, and we found no evidence for resistance at that stage. So that was back in 2006, 2007. What we've been doing since that time is continuing to, I guess, get a handle on what is going on out in the field regarding resistance. And as part of that work, we've actually been screening a number of different chemical compounds. And I guess this is just a quick snapshot of some of those results. And I guess the news is perhaps somewhat good, somewhat bad. If we actually look at a number of other chemical groups besides the SPs, so organophosphates, carbamates, fipronil, and a few other of these newer chemicals, we actually have not found resistance to any of these chemical groups out there in the field, which is good news, which means we can implement some insecticide resistance management programs, and it means farmers still have options to control mites even if they are resistant to synthetic pyrethroids. However, what we found, interestingly, was within the entire synthetic pyrethroid group, when we looked at both type 1 and type 2 synthetic pyrethroids, we found resistance was across that entire group. So I guess the message there would be if the farmer's got a particular instance with resistance to a synthetic pyrethroid, there's no point in grabbing a different synthetic pyrethroid. You really need to get out of that chemical group. Svetlana is going to come up next and talk a little bit about, I guess, the geographical spread of this resistance within WA. At this stage, it appears to be confined within WA. We are doing some screening on the eastern states, but at this stage we've not been able to detect resistance over in Victoria and New South Wales. But I guess what I just wanted to finish with these 2 slides to really, I guess, bring this particular case study back into the broader scheme of things around, I guess, pest management in the grains industry. Unfortunately, this resistance story is not unique. It's not a new phenomenon, and there's a number of other very important broadacre pests that have already developed resistance to, in some cases, multiple chemical classes, and also in some cases that resistance is quite widespread across Australia. So we've got species such as Helicoverpa, diamondback moth, those sorts of species that have developed resistance, and I'm sure many people again are more familiar with resistance issues in some of these other species, such as sheep blowfly, grain storage insects. I'd also just want to make mention that again we've seen a recent development last year in 2010, Again, this was some work done by the Department of Agriculture in conjunction with CSIRO, but they uncovered for the first time within Australia the first cases of pyramicarb resistance in green peach aphid. So again, we've got this theme of potentially species finding a way to overcome our current, I guess, pesticide-based management strategies. So my last slide before Svetlana comes up is to really just leave you with a few take-home messages. I guess the key one there is that the resistance that we're seeing within red-legged earth mites is likely to increase. There's no doubt populations out there already that have resistance that we haven't uncovered. So I guess some management and take-home messages here would be that we really need to, I guess, think smarter about when we're applying chemicals, spraying only when necessary, only when we're seeing damage, and we really need to get away from that insurance spray mentality. So throwing in a cheap SP when we're running around a paddock with a herbicide, for instance, certainly common practice back home in Victoria. It is important to get our identification right. As I mentioned earlier, red-legged earth mites are often mistaken for other mites such as blue oak mites and Belorstian mites and Bryobia mites, and they actually do require slightly different management strategies. So it is important to get your IDs right. There's things that we can do around weed management, so we can control weeds both within a crop and also along fence lines as a way of reducing the habitat that red-legged earth mites like to survive in. As I mentioned, some people are already practicing these things, crop rotations and pasture grazing management as a way of reducing mite populations, and that negates the need altogether of using chemicals. And of course, now that we have this resistance, we need to think carefully about rotating our chemicals. So, you know, growers and advisors should get a good understanding of resistance management strategies, and certainly there's some great lessons to be learnt from the weed resistance issues and the herbicide resistance issues in weeds, I should say. And I guess the final point that I'll leave you on is that we should be going back and inspecting spray coverage where possible. Don't necessarily, I guess, assume that a chemical that has been applied will work effectively. So I'll just pass you on to Svetlana. I'm more than happy to answer questions at the end.
We've learned how to use this. Okay. Basically, we found the first lot of resistance in 2006 in Esperance, but CESAR and DAFWA did testing in 2007, and what we found was that, oh, there it is, okay. Esperance and one site at Cranbrook are the only sites that had a spray failure in 2007. We had reports of another 2 spray failures, which were we found were spray application errors, one at Manjimup and another at Jaramunga. And we did some random surveys and found another 2 sites with resistance. So that just shows that resistance in 2007 was limited to 2 sites which had crop damage and 2 sites which were part of just random resistance testing. Right, go back one. In 2008 we did some more resistance testing. In 2008 I had a grower in the South Stirlings region who reported spray failures and another grower at Manjimup. The grower at Manjimup did not come up with resistance. It was again a spray application error. And the grower at the South Stirlings had to reseed. He had extensive damage to his crop. But what you can also see is that there's still quite another 3 sites there with no resistance, so it's still quite patchy. In 2009 we did extensive surveys of resistance between Perth and Albany, and we found no sites between Perth and Albany in those blue with resistance. The sites that we did find with resistance were sites that people came up with saying that they'd had spray failures, and there were sites again at the South Stirlings and Woodjanilla, not this year. But you can see that it's still quite patchy and not very widespread. In 2010, we basically looked at resistance surrounding properties. In 2010, I had 2 growers that reported spray failures, one from Woodjanilla, another one from two from Esperance and 2 from South Stirlings Woodjanilla region. The others that you can see on the map are all the result of random resistance testing that was done in conjunction with agronomists. The agronomists targeted growers that they knew had not rotated their chemicals and were likely to have resistance. So they were targeted, and we found, as you can see from the red stars, a whole bunch of sites where there was resistance. You can see that the resistance is patchy. It's only in certain areas. So we've just got one site at Esperance, sites around the South Stirlings, Cranbrook, and one site at the Tenterton region, and there was one outlier here at Boyup which is also a spray failure site. It does show that we do have a lot of gaps to fill in, and we will be doing resistance testing in 2011. So if you do happen to have clients, or you would like to have properties tested, we will be offering that service. So please contact Peter Mangano if you happen to be in the northern to central ag regions, or myself if you're in the southern ag regions, and we will arrange to have resistance testing done. We'll say at this stage we've had issues with growers not wanting their properties tested, mainly not wanting to know, or putting in sprays like OPs and things like that, just in case they have resistance, because they haven't wanted to face that fact. So that's something we're slowly overcoming.
So high populations of invertebrate pests will reduce your crop yields and the storage quality of grains, resulting in usually heavy economic losses. Designing a sustainable strategy to control or manage pests will be essential for the future of the grains industry. Presently, the development and promotion of an integrated pest management program for broadacre farming lags well behind that of other industries such as cotton. However, ongoing development of grains IPM programs have now become even more difficult following the validation of resistant populations of red-legged earth mites, aphids, and diamondback moth in parts of Western Australia. To effectively combat insecticide resistance and manage pest outbreaks, we must first understand the complexities of pest insect biology, the relationships which have developed between the pest and the plant, any interactions likely to affect the spread of pathogens carried by these insect pests, the susceptibility or resistance of a crop to attack, and the external stresses driven by the environment which cause the crop and a pest to change. To do this, GRDC recently funded our project developing and promoting IPM in Australian grains, which aims to benchmark current pest management practices within the industry and examine reduced insecticide approaches to insect pest management in grain crops across all of Australia. After 2 years of this project, growers will start to gain a greater appreciation of alternative pest management tactics and how these tactics might be used as part of an integrated pest management approach on the farm, with the hope of reducing Australia's reliance on broad-spectrum insecticides. In winter 2010, a large consortium of researchers and farming groups established 5 on-farm trials to address the uptake of IPM in broadacre farming. Canola was sown across all of our trial sites in the 2010 season, and this year we'll be assessing wheat at the same sites. At each of the trial locations, a series of 12 plots, each greater than 50 by 50 meters in size, were assigned to one of 3 pest management approaches. A no insecticide approach, which was used as an experimental control, an IPM approach that involved monitoring pest and beneficial invertebrates, combined with plant damage assessments and the application of softer insecticide sprays, but only when needed, and a conventional approach based on the current farmer practice to manage pests in the local region. Invertebrates were assessed in all of the plots throughout the duration of the growing season using a combination of collection and assessment methods. These included vacuum sampling, pitfall trapping, direct visual searches, sweep netting, and extracting invertebrates from soil cores. In addition, plant-based assessments were also conducted during the establishment period. These included plant density counts and measures of the level of plant feeding damage to the plants themselves. We also had dry matter cuts of plants taken prior to harvest, and then these were threshed to calculate the harvest index value in order to compare harvest yields across the different locations. And at the end of the growing season, crop yields were measured with a 1 meter wide plot harvester, and in some cases by 10 meter wide machine harvester. We still have a large number of invertebrate and plant samples which we are processing. across each of the states, including WA, so the results discussed here today must be considered preliminary only. In Victoria and elsewhere, the IPM treatments incorporated an insecticide seed dressing, while the conventional approach received 2 separate foliar sprays, usually a bare earth and a post-emergent application. After crop emergence and up to 21 days, as shown on the graph, there was a significant reduction in plant numbers in the control plots. There was no difference between IPM and conventional plots. Harvest index values also indicated that canola plants in the controls produced more seed per plant, total plant biomass, and this was likely due to early season insect damage or environmental stresses, lowering the density of the plants, resulting in less plant competition at the end of the season when there was very, very high rainfall and very low pest pressure. Canola crop yields at our Wickepin trial site in Western Australia showed a similar trend to the other national sites. Overall control plots were different from the IPM and conventional plots, but there was no major difference we found in the yield between the IPM and conventional plots. The canola crop yield at our Beverly site showed an even stronger difference between controls and treatments, but again, there's no major difference between IPM and conventional treatments. The harvest index values at the Beverly site matched the corresponding crop yield data. As expected, control plots had a lower harvest index value than either the IPM or conventional treatments. So overall, these results suggest that crop yield and harvest index do not differ between an IPM approach and a conventional approach. And so I guess we're led to why would a grower adopt IPM? Well, we find that in drought years, at least here in WA, when profit is tightest and crop yield is low, growers will probably need to be more conservative with the cost of their control sprays. And it's also possible that growers may apply more chemicals to protect what they've got remaining, to protect the reduced yield during drought years, which, as the data above shows, no additional gain would likely be achieved. Preliminary cost-benefit analyses from our Victoria site indicates that a conventional approach may cost a grower approximately $11 per hectare, while the IPM approach may cost approximately $1.35. So given that IPM is an accepted approach to sustainably and cost-effectively manage invertebrates in many other countries, the grains industry in Australia will need to investigate the cost of the monitoring and the time taken to identify invertebrates, as these are components these parts of these components are likely to be an ongoing challenge in broadacre farming, particularly for the larger farms that we have here in WA. And we're going to consider this in our 2011 assessments, and hopefully get back to you next year with our updated results. Lastly, if you recall, one of the aims of this project is to benchmark current pest management practices within the industry. And to do this, we'd like all of your assistance in participating in our IPM survey. Surveys can be completed online via this website, and there's also paper copies available at the back on your way out if you want to pick one up. So thanks everyone for listening to our presentation today. Svetlana, Paul and myself will be around to answer any further questions.
I just wondered, Svet, what you thought, why it was quite concentrated right down there in the south, the resistance. I just thought that area around Catania, etc. would have had a few come back as well.
It's probably where the spray failures come from. It's down south. It's also where we have the highest pest pressures. The survey really reflects where we've actually looked and where we've had reports of spray failures. We have looked around Catania, but we've only had reports of spray failures no one around Catania has actually reported a spray failure. I suspect that there is resistance, but they're keeping quiet about it.
And I might direct this question to both yourself and Paul, because you're both involved in this space, but with the figures you put up, the $1.35 a hectare for the IPM cost, I assume that was the chemical treatments used within those operations. Have you made any estimates of what the actual monitoring costs might have been, either in the WA trials or perhaps Paul, what they've been in Victoria?
We currently haven't done any of the cost-benefit analysis yet for any of our Western Australia sites, but we do plan to include that in our 2011 assessment. So I'll hopefully have some of that information for you next year.
Yeah, just to clarify that this data came from the Victoria side. Essentially, the reason the IPM approach last year was quite low at $1.35 was because the IPM treatment solely had a gaucho insecticide seed dressing at the start of the season, and therefore there were actually no applications, and it was a very low pest pressure year, so there were actually no foliar applications the entire year needed on the IPM plots. Whereas with the conventional approach that had a bare earth application, although the chemicals are still reasonably cheap, the application costs add up to that $11 a hectare. It had a bare earth plus a post-emergent foliar application as well, which is essentially the conventional practice within that region. Regarding the question from our end about have we started to factor in from an economic point of view how much time and therefore how much expense is involved in monitoring and the sorting of samples, we haven't done that. And I guess that's part of the challenge with this project, because we're still not sure, and I guess Laura didn't have time today to go into the details of the methodology, but we're actually doing two types of sampling. We're doing one where we're really trying to generate some very scientific, rigorous data that we can go out and to present to people, but we're also doing some what we're calling demonstrative sampling, which we think is what a farmer might be willing to do. It's far less. So for instance, we're doing more just direct counts. We're not doing vacuum suction counts and all those sorts of things. So the issue, I guess, at this stage about trying to get a good handle on how much time is spent in the monitoring is that we actually don't know how much monitoring we need to do. So that's hopefully something that will, I guess, play out over the next year or so with these trials.